a social media editor for the journal Cannabis and Cannabinoid Research, and also here today helping with the UC Irvine's impact of cannabinoids across the lifespan. I am very excited to be interviewing the director of ICAL today, Dr. Daniele Piomelli, and we get to hear a little bit more about him and his work and his vision for the center. So Daniele, can you start by introducing yourself and tell us a little bit about this center?
Thank you, Hilary, for doing this. So my name is Daniele Piomelli. I'm a distinguished professor of anatomy and neurobiology at the University of California, Irvine, and I'm also the director of the P50 Center of Excellence funded by the NIH NIDA called Impact of Cannabinoids Across the Lifespan, Impact of Cannabinoids Across the Lifespan, which, as the name implies, studies how cannabinoids, and particularly tetrahydrocannabinol THC, can affect the brain and body of an adolescent in ways that are either very persistent over time or even permanent. And we use animal models. We use rats, mice of both sexes.
Fantastic. So what inspired you yourself to get into this specific research area?
It was the legalization of cannabis, which started, I am in California, in Irvine, California, and in our state, cannabis became first legal for medical purposes in 1996, and then it became fully legal in 2016. So now is quite widespread, and I'm fine with that. I don't think that that's per se a problem. But what is a problem is that we don't know enough about many of the things that cannabis does. Particularly, we don't know enough about how it impacts certain vulnerable populations, including teenagers. And I'm going to add persons who expect, and also people above a certain age, 65 or 70. So that was fundamentally for me kind of a public health kind of motivation that jived with my prior interest in cannabinoids and endocannabinoid pharmacology.
Right. So bridging your background in more of the nitty-gritty science with a really emerging public health problem. Yeah, I'm here in Michigan, and same thing. We legalized cannabis for recreational adult use a couple years ago, and you can't drive, 5 minutes on the expressway without seeing a big cannabis bulletin. So certainly the public opinion has outpaced the science in terms of cannabis. So
Yeah.
super important. Can you talk a little bit more about this P50 center? Like, what is the overall mission?
So the National Institute on Drug Abuse funds this sort of cooperative centers. They're called Centers of Excellence, or P50 is the bureaucratic term for them. And the purpose of these P50s is always to move forward a particular field in a way that individual investigators could not move forward, could not do it. Individual investigators with grants can do excellent work, sometimes fantastic work, but the problem is that they're limited to one specific topic, one specific problem. And what centers try to do, and ICAL in this is organized to do that, is to bring together multiple investigators, look at a problem from different perspectives, and try to move forward the field more effectively or more rapidly, if you wish, than if we were all separate and working independently. So in this case, I'm very lucky that we are UC Irvine has a very strong neuroscience community, and I found a small team of investigators in the School of Medicine and School of Biological Sciences who were interested in topics related to cannabinoids, but not quite cannabinoids, but they were interested in applying their knowledge on their specific topics to endocannabinoid science and cannabinoid science. So we teamed up and put together what was, this was 6 years ago, a fairly compelling application, which was eventually funded. And we did the first 4 years of work and then wrote a competitive renewal. And now it's been funded again for another 5 years this time, and hope to be able to complete the work we started 6 years ago.
That's fantastic. And yeah, bringing a true kind of interdisciplinary and team science approach to this really important public health issue definitely strengthen numbers and working together instead of, like you said, there's only so much we can accomplish as individual investigators. So I'm also very excited to see what this latest renewal produces. What would you say is really unique about ICAL?
I don't know of any other centers that do what we do. very few centers are interested really in understanding impact of a drug class in a specific time of development and look at the effects of this of the exposure to the drug over what in the life of a mouse or a rat would be the equivalent of human decades or many years. So I think that is unique. The other unique, well, maybe not unique, but certainly the other distinctive characteristic of ICAL is that we bring together folks like me, who is, I mean, I'm fundamentally a pharmacologist slash neuroscientist, but I've done pharmacology all my life, with electrophysiologists and behavioral pharmacologists, people who have, again, very different outlooks on the same problem. And I have to say that this has been really, for the first 4 years we were funded, this has been extremely productive. And some of the discoveries we made, I don't think we could have made if I had been working by myself and the others had been doing the same. So I think this was where a place where synergisms really made a qualitative impact on the outcomes.
Yeah, that's fantastic. And I'm also a developmental scientist working more on the human side of things, but I think a lot of people just think that you can study, you know, cannabis effects on the adult brain and extrapolate that down to adolescents, but, you know, they're not just shrunk down versions of adult brains. It's an entirely different system. So I'm just really glad that your center is really focusing on that unique, you know, physiology and biology of the adolescent brain.
You know, we've been learning so much. One of the things we learned is that whatever we do in adolescence and then we repeat in adulthood, the same exact treatment regimen, completely different effects. So clearly, this is a system that evolves with time. The body evolves with time, naturally, but this is also a system that evolves with the body. And when you affect it, you know, at different ages, you have different results.
Right. Yeah. And I want to get to more of like the planned and current projects, but you're just making me think of some of your recent findings from your center, which highlight exactly that. Could you talk a little bit more about some of the recent findings from ICAL?
Oh, it's a lot of work. So maybe because one or two, we really spent a large amount of energy on this particular topic. So, but maybe one thing I could point out is kind of weird. It's something that, you know, I found interesting because it's a little bit weird. I'm not sure that everyone will agree with me. But one thing that we discovered is that if you treat mice or rats with THC, the active principle of cannabis, in adolescence, one of the structures in the body that is affected the most is, even before the brain, the adipose organ. So white adipose tissue, brown adipose tissue, the stuff, the organ where we store energy, and it actually bothers most of us because we've got too much of it. That particular organ is profoundly affected and permanently affected by exposure to THC in adolescence, only in adolescence. Because if you do the same treatment in adulthood, nothing happens. And it's actually kind of striking. Let me give you one technical detail. If you give THC in adolescence, you pull out the adipose tissue and you look at the genes that are affected, you know, the expression, the transcription of which is affected by THC, we're looking about 2,500 genes going up and another 2,400 genes going down. So we're about 5,000 genes changing. That's 20% of the rat genome. If you do the same experiment in adulthood, exactly the same thing, you have about 40 genes going up and another 20 genes going down. So you have a tiny amount, tiny fraction. What that tells us is that when you intervene at a time where an organism is in a crisis, and by crisis I mean a situation where it is facing hormonal in waves, and the hormonal landscape is constantly changing, and the animal is undergoing this enormous growth. In those critical times, intervening with a drug, and I think THC is a good example, but the other drugs have similar effects or other effects, but intervening with a drug can have very profound effects that last way more and the other than expected. And the other thing that I found exciting about this finding is that, and I think I'm the only one here, is that it doesn't involve the brain. And that's another thing, that when people think about cannabis, because you smoke it to get high, so clearly you're thinking that the brain is the place where it goes, and that's all. That's the end of the story. But in reality, it's far from it, because THC goes everywhere in the body. We have cannabinoid receptors pretty much in every single cell type in our body. We have an endocannabinoid system that is ubiquitous or quasi-ubiquitous. So there are organs like fat that become affected even though you don't know, and you don't expect, you don't imagine them to be affected. And so one could say, okay, well, whatever. So who cares that the fat is affected, right? So you have all these genes. Who cares? Well, the thing is that the problem is that the fat stops working properly in adults that have been treated with THC. In adolescents, the fat doesn't work properly, and that means that when you need energy, because energy comes from fat, most of it, it comes from sugar that we eat. But if we use the sugar, then we need to recruit energy from fat. When you need the energy, you don't have it. And that is a problem with the brain, because our brain needs the energy, something called ketone bodies, to be able to work, especially under conditions of stress. So I feel that when we look at the problem from different perspectives, not always the same thing, I think we can make interesting and unexpected discoveries.
Yeah, that is so interesting data. And I think a lot of people will understand that like adolescence is a sensitive period. A lot of people that use cannabis begin using during adolescence. And what your team was finding is that there's really unique impacts that, could have effects on the whole body and the whole brain. And as neuroscientists, we need to be thinking about things, I guess, other than the brain, right?
Well,
Or
the brain is in the body. It's annoying maybe for some neuroscientists, for many, maybe for most neuroscientists, an annoying fact. But it's not. It's not an opinion. It's a fact. It's in the body. All it does, it feeds off the body and controls the body. So you can't imagine any action that impacts the body that does not impact the brain, and vice versa.
Yeah, absolutely. If it requires what 30% of the energy we take in goes directly to power up here, it's
Yes,
enormous.
yes, it's about 1.5 kilograms, 3 pounds, and 20% of the energy of the body. So it's about 1% of the body weight and about 20% of the consumption of energy consumption. Yeah, it's a big deal.
Yeah, so you guys have done a ton of unique discoveries in this last period. So tell us a little bit about the projects planned for the coming couple years.
Oh, great. So that requires me going to a slightly different project, different set of discoveries we made. Uh, so we don't just look at fat. We also look at other tissues. So in particular, we do look at the brain. And there's one cell type in the brain called the microglia. And microglia are a very interesting cell. They're immune cells. They are not uh, uh, they've been studied for many years, known for many, many years. They were discovered by Ramon ICAL, I believe, or one of his students back in the beginning of the 19th century, of the 1900s. But only in the last maybe decade, 5 to 10 years, they've become really interesting to a lot of neuroscientists, because we are now starting to appreciate how critical they are, even to things that we always thought were run by neurons. For example, the response to social stress. The response to social stress is something that is super important in everyday's life. Some of us don't respond well to stress, others do better. But social stress for us, hypersocial animals, is critical, because we need to be able to learn how to cope with the difficulty of interacting with others. And also, uh, we develop protective mechanisms when our our behavior brings us to a challenge, and we are challenged by the sociality of our behavior. So that sort of that negative feedback is something that we could experience. For example, when we are defeated, right? So let's say that okay, here's me an example that is valid for both of us. We go on the podium, we are invited to give a lecture. It's super important. Maybe even a job is dependent on it. So we are there and we are very nervous. We go there. So first of all, why are we nervous? We are nervous not just because maybe a job is depending on that, but also because we're speaking in front of like 50, 100, 200 people. And so that is already social anxiety that comes as part of the social response. And let's say that we are defeated in the sense that I gave a very bad presentation, so I go home and I feel like crap, that everything has gone wrong. So that kind of social defeat has consequences if it happens in certain particular time of your life, for example, during adolescence again, or it happens many times over your lifetime. Then you could develop things like depression, like social anxiety and disorders of this type. So the microglia, I'm sorry for this very long.
Oh, this is helpful.
The microglia are very important to this response. That's weird because you wouldn't imagine an immune cell to respond to that, but in fact, it's a complicated network that starts in the periphery, goes to the microglia. There are immune cells in the periphery of the body that talk to the microglia in the central nervous system. They talk to one another, they talk to neurons, and at the end of the day, you develop a response. So it turns out, and I'm sorry for this long introduction, but it turns out that if you treat animals with THC, this is both mice and rats, both males and females, if you treat them with THC during adolescence, the microglia change for a large portion of their young adult lifetime. They become less responsive, less capable of becoming activated by socially stressful events, or by, say, microbes, microbes like in bacteria or in viruses. So they become less responsive, which means that when they are stimulated, they cannot mount a response that is normal, that is physiological. So now someone, again, this is an area where people come in with a lot of expectations. And sometimes, they should set expectations aside and just look at the data and try to understand what the data are telling us. So some people react to this and say, okay, wait, so it's good. It's a good thing because THC is now making the microglia less responsive to stress. So you do better because you're less responsive to stress. You're not so socially awkward, for example. You don't feel bad when you have a social defeat. The problem is that if you take a cell that is normally meant to do a series of things and you change it in ways that are so profound that it cannot perform its normal job, then you almost certainly must expect that some of the normal physiological actions of this cell now are compromised. And that is, I think, exactly what happens. I think that we don't know about humans, but animals that are exposed to THC in adolescence, when they grow up to become adults, their microglia does not work well anymore. And this means that their interaction with neurons is not physiological, becomes quasi-pathological. I don't know if it's really pathological, but let me say it's certainly not normal. And so this is where we are heading now. In the first 4 years of our grant, we identified this profound change in the microglia. And now we would like to understand how this change affects the neurons and the other cells. And by doing that, how it affects brain function. And let me just say one more thing. So one of the things that we discovered, and by we now I mean the electrophysiologists in our ICAL team, what they discovered is that exposure to THC in adolescents has a very profound negative impact on a form of synaptic plasticity in the hippocampus called long-term potentiation, and particularly in one form of long-term potentiation that is very important for to encode episodic memories. Episodic memories are those that relate to your life. You think, when I was a child, my dad and my mom did this and that. Those are all episodic memories, and those are fundamental to human life. Episodic memory is the first that goes with Alzheimer's disease. And the issue that these animals have, they don't encode episodic memories anymore. Their LTP is compromised, and they don't encode episodic memories very well anymore. So we suspect that this is due to the fact that the microglia cannot talk properly to the neurons. So the neurons either don't develop properly, or maybe the fact that the microglia is not producing, I'm just suggesting things, producing the right type or the right soup, if you wish, of mediators makes the neuron unable to work properly. So it could be either kind of a developmental effect or a more tonic effect on neural function. Because this is where we are going with this hypothesis.
Wow, that is fantastic. And I can't wait to see what you guys find out. And I think for people that aren't in our field, they just don't appreciate how cutting edge everything that you're saying is. I mean, understanding, like you said, the critical role of microglia in brain functioning and brain development and things like learning and memory and response to stress and just implications of THC exposure. So this is super cutting edge and novel things. And I could see a lot of directions that this research could end up going very broadly. So I'm just very excited.
Well, thank you. Just to add a few things about what you just said, I think the real scientific innovation is always at a crossroads. It's always at a place where you have one area of research and another area of research that slightly overlap. In this particular case, for example, one of my colleagues at ICAL, his name is Professor Kim Green. He is an expert in microglia. He is not really interested. He wasn't really interested in cannabinoids before I started. I converted him. Okay, I brought him into the fold. Anyway, so one thing that we discovered really is that it was completely surprising to him as well. Is that you could have this profound effect on microglia with a drug he hadn't even thought about before. So that is exactly to your point, is that when you bring together different areas and you make the investigators talk to one another, like as it happens in a P50 Center of Excellence, new stuff comes up, new things that that were unexpected before. Otherwise, we're always talking to ourselves. That's a problem we scientists often encounter.
Yep, and we scientists sure like to talk about our science.
Sure do. And you're indulging me with this. Thank you.
Oh, it's fascinating. I could do this all day, trust me. So how do you see the science coming out of the center, either informing public policy or really that public conversation about cannabis?
Understanding better how THC can affect an adolescent is critical. Because call me naive, but I believe that you got to tell people the truth. And then they do what they want with it. And this starts when they are, when people are young, because I think at 15, 16, although of course our brain is not completely developed yet, one has all the, the ability of understanding arguments and evaluate data. I think that if we understand exactly what are the risks that one encounters when one uses cannabis in adolescence, we can achieve at least two things. One is to tell the truth to people instead of saying, "Okay, oh, this is your brain on drugs," and showing a frying egg, which is bullshit and never works, right? Because people then have their friend, they know that their friend is just fine, and they say, "Okay, I'm not going to believe anything that comes from this propaganda." Giving people the scientific data to the best of our abilities is, I think, one of the reasons we're doing this at ICAL. The other is really because we need to understand better where the risks are. And we don't really do that. We're not very good at that. And by that, I mean, again, if you throw in a blanket and say, "Oh, THC is the most dangerous drug on earth," as you still see today. I mean, you see investigators making this type of statements, blanket statements, that are just not supported by the data. If you say something like that, no one is going to believe it. It's going to be a counterproductive discourse. You're going to get people to ignore you, whereas there are real risks. So I believe that if you understand what those risks are and you define them precisely, as opposed to these blanket statements, then people, again, we go to the previous point, people can make a rational decision. Because people need to make rational decisions, but they need data to make the rational decisions. And this is what I think something like us, the P50, and me as a scientist, I really believe this is one of my roles as a scientist, is provide those data to society. And so really two different things, but I feel that they're also obviously interrelated.
Yeah, absolutely. And also, you've got a moving target because, like we were alluding to in the beginning, there's a changing legal landscape, there's changing public opinions. Cannabis today is totally different than cannabis 20, 30 years ago. So the data that you're culling right now and you're researching right now is more up to date than a lot of the older studies. And so there's a lot of challenges as well. But I guess from you, what is it that excites you most about this changing field?
Oh, there are quite a few things. You know, the motivation to do the work, which I just shared with you, is really not the same as the reason why I'm happy to do it. Because the reason why I'm happy to do the work, I come to work, whistling opera instead of being, grumpy, etc., is because there are some really interesting things that I feel that we need to understand. And the endocannabinoid system, THC, whatever, these are all windows that allow us to open, on this gigantic landscape that is the brain, and allow to illuminate the brain in one perspective different from another perspective. So create some true new knowledge. So the things that I feel are most exciting about the endocannabinoid system right now, and that, that for our listeners, because the endocannabinoid system, maybe they believe has been known for 100 years. No, it was actually discovered between 1992 and 1994. The very first papers were published back then. So it is 30 years old, but it is not 30 years in science are not a very, very long time. So there are many, many questions that are still open. And one of the questions that I'm most fascinated about is that it's not a system that operates in isolation. Is a very interesting system in that it uses um, the components of the cell membrane, which are lipidic, so they're fat-like components, and it uses these components to generate signals. So in a sense, is unique because most transmitters in the body are not derived from the cell membrane. They are derived from their amino acids or biogenic amines. So they're pretty much water-soluble compounds. Instead, the endocannabinoids are not water-soluble. They come from the lipid membrane, and that is interesting because they're not by themselves. They are part of very complex interacting systems that share similarities with the endocannabinoid system, but they're different enough that they're not overlapping with it. So they have some sharing and some non-sharing, and we call this novel mediators, we call them paracannabinoid, P-A-R-A, paracannabinoids, to mean that they are related to the cannabinoids, but they are different enough. So para in Greek means around. So they are working together with the endocannabinoids. They work together, sometimes facilitating what the endocannabinoids do, sometimes inhibiting what the endocannabinoids do, and but they're very, very important in modulating the responses to the endocannabinoid. So this is one area where we're spending a lot of resources, a lot of time making interesting observations. So that's one area that I'm excited about. Another area where I've been excited for quite a long time, and it's been also a thorn in my back because it's extremely frustrating, is the development of new medications based on the endocannabinoid system. So the endocannabinoid system offers a variety of potential targets for drug discovery. There are enzymes, there are receptors, there are transporters, all these various molecules. There are proteins, of course, so they can be targeted by a small molecule. You could design a small molecule to go and modulate a particular function. So we've been trying to do that for a long time. We've succeeded on paper. That is, we succeeded. We can cure mice, we can cure rats, but one thing that would be for me really important to see before I'm no longer around, is to see that at least one or two of these things has been tested enough in people to be able to say, okay, it worked or it didn't work. So that's the thing. I'm not espoused to, okay, I don't want to necessarily see something to work because that would be lucky. But I would like to see some of these ideas at least to be properly tested so that we can say, okay, I've spent this many years working this out. It works in the rat, works in the mouse. It does not work in people. And that's fine. It's fair enough. I don't work with people. So what am I going to do, right? But that's something that is frustrating me. But I do hope that in the next 5 to 10 years, we will be able to get to a point where we'll be able to answer some of those questions.
That's fantastic. Yeah, and in your description of this crazy new area in modern medicine and science, I think you're highlighting for young students or researchers who want to get involved that the landscape is wide open in terms of discoveries for understanding just basic neuroscience and biology and physiology, as well as potential treatment implications. So
it is absolutely, you're absolutely correct. It's still completely open. I want really the students to appreciate this because when you come in as a student, and of course you don't know anything, why would you? I mean, that's why you're coming in as a student. You look at what the body of knowledge, and sometimes you can feel overwhelmed and you think, okay, how am I going to find my own spot? The reality is that there's so much to be discovered in every field, including the endocannabinoid field. I mean, I always say to my students, I used to say when I taught pharmacology, this is what we know about pharmacology, and this is what we don't know about pharmacology. There is a lot more than we don't know that we do know.
yeah.
The only trick, and that is a big, big trick, is that you got to be able to think a little bit outside of the box. If you as a student, you want to fit in, right? It's like, you want to be part of a crowd, you want to be part of a team, whatever. But that is only good. They only will take you so far. What you can do that, but what you most importantly need to do is to just try not to think like the same group that you want to be part of. You see what I'm saying? Is a little bit counterintuitive, but that is really what is important in science. Yes, you need to fit in because science is a social effort, but you also need to be out so that you can actually look at the field from with an outside view and say, okay, you know what? I need to do that, and no one is doing it. One good way of doing this, actually, this is a piece of practical advice to anyone who is interested. Yeah, the cannabinoids and endocannabinoids is a great field, blah, blah, blah. But try always to go somewhere else and learn something else and see if that applies to the field you're most interested in. So one reason I was lucky enough, and I had huge luck in my life with this, to make discoveries on the endocannabinoid system early on in the 1990s. We discovered how the endocannabinoids are produced and how they are degraded. We made the first inhibitors, etc. The only reason why we could do all this work is, well, I did work my ass off. But aside from that, the real reason is because I came from a different field. I was in the field of inflammation. I was studying eicosanoids, and I arrived in the field of cannabis when anandamide had just been discovered, and everyone was like, "What is this thing?" So what? And they said, "Oh, look, this is like one of the eicosanoids, only different. It cannot be produced this way. It has to be produced some other way." So that is how discoveries are made. You come in from one field into another field, and you try to inject the mentality and the knowledge of the other field into the receiving field. I don't know if I've been clear, but I feel that's the only piece of useful advice that I can give to anyone, even your age, Hilary.
I think that's perfect. And your ICAL really exemplifies that because you knocked on the door of a physiologist and you said, hey, come take a fresh view at these questions. So that's fantastic. I have two shameless plugs on a follow-up to that. If you want more information, Dr. Piomelli is the editor-in-chief of Cannabis and Cannabinoid Research, which is the very greatest cutting-edge publication in the cannabinoid space. So definitely check out that publication journal. And then the International Cannabinoid Research Society, or ICRS, is a fantastic conference that Dr. Piomelli is affiliated with. Do you want to say a few words about that interdisciplinary conference?
Oh yeah, ICRS has been around for a while, and I think it's a great place if you're a student, if you're a postdoc, to meet people. I think pretty much the entire cannabinoid and endocannabinoid field meets there. Not 100% of the researchers, but I would say a good 70 to 80% of the researchers do meet there. It's a very friendly, amicable environment. People are not stuffy, and they talk to one another a lot. And our next meeting is going to be in Bloomington, Indiana. So anyone coming to the meeting will have nothing to do but be at the meeting. So this is spoken like a true Californian. I'm
I'm
sorry,
in the Midwest, but I totally understand. Yep.
but it's good. That's what you want to do at a meeting. You don't want to go to a meeting to be a tourist. You can be a tourist after the meeting or before the meeting, but during the meeting, you want to be with the people of the meeting. You
It was
invest in it.
intentional. I see. Okay.
That's, that seems like the right thing to do.
That's perfect. Fantastic. So to find out more about the center, we are on social media now at UCI Cannabis. Dr. Piomelli, is there anything else you'd like to share with us about your center or the vision before we wrap up?
No, thank you so much. Thank you so much, Hilary, for
Okay.
this.
Thank you. I appreciate it. Take care, everyone.